Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture series on special products from amino acids, I am going to concentrate on formation of histamine and gamma amino butyric acid. Now let's look into details on formation of histamine and gamma amino butyric acid. Histamine is a biogenic amine and it is made from the decarboxylation of histidine by histidine decarboxylase enzyme. And as I previously said in my earlier lectures, that whenever amino acid is undergoing decarboxylation to make a special product, so the amino acid decarboxylases, they need pyridoxal phosphate as the coenzyme. So same thing applies here in the conversion of histidine into histamine. And what is the role of histamine? Histamine is a major mediator of allergic reactions and it is synthesized by the mast cells and it is involved in gastric secretion and made from basophils and mast cells in our body. And once histamine is degraded, so it will be inactivated into methyl imidazole acetic acid and indole acetic acid. Now let's move on to see the functions of histamine. One of the major functions of histamine is it acts as a vasodilator and it is going to increase the capillary permeability. Histamine also acts as a smooth muscle contractor. It contracts the smooth muscle, especially in the branchial and intestinal smooth muscle. And over gastric mucosa, it is going to increase a gastric acid secretion and also in the nasal lining, it's going to increase a nasal fluid discharge. So overall, as a response to allergic reaction, there will be branchial contraction, intestinal smooth muscle contraction and increase in gastric acid secretion and nasal secretion. And also histamine, it is going to regulate immune system. So overall, the histamine's action will be conducted by histamine binding to two kinds of receptors. One is H1 receptor, other is H2 receptor. Histamine 1 receptor and histamine 2 receptor. Now the histamine 1 receptors are found over smooth muscle cells like branchials, smooth muscle, blood vessels, smooth muscle, intestinal smooth muscles. Thereby their action on the increase in the capillary permeability, smooth muscle contraction, intestinal contraction, all of them are mediated through H1 receptor, histamine binding to H1 receptor. And that effect of histamine binding to H1 receptor is counteracted by H1 receptor blockers, and two drugs that I have mentioned here which are H1 receptor blockers are diphenhydramine and chlorpheniramine, usually coming in as chlorpheniramine maleate. So the diphenhydramine and chlorpheniramine are H1 receptor blockers. And over gastric mucosa, H2 receptors are the ones through which histamine is working. And we have H2 receptor blockers which are used in the treatment of gastritis, increased gastric acid secretion. And example for that is cimetidine, ranitidine. So these are H2 receptor blockers which will decrease gastric acid output. Coming to formation of gamma amino butyric acid. Gamma amino butyric acid, like in the previous slide what we have seen with conversion of histidine into histamine, here glutamate undergoes decarboxylation to make GABA. Likewise, there is a requirement of pyridoxal phosphate, just like as I said in the previous topic, that amino acid decarboxylases, they need pyridoxal phosphate as the coenzyme. Now, what is the function of GABA? GABA is a major inhibitory neurotransmitter in the central nervous system and defect in the GABA decarboxylase enzyme, where it will lead to decrease in the availability of GABA, which is an inhibitory neurotransmitter, will lead to imbalance in the inhibitory to 𝐒𝐧ℎ𝐼𝐷 neurotransmitter. When inhibitory neurotransmitter levels goes down, like GABA levels decreases because of defect in glutamate decarboxylase, will lead to increase in the activity of excitatory neurotransmitter, that is glutamate itself. Glutamate is an excitatory neurotransmitter along with aspartate. So that may lead to convulsions, chorea, because the seizure threshold will decrease when GABA decreases. And this seizure or convulsions can also be because of decreased pyridoxal phosphate. Vitamin B6 pyridoxal phosphate, it is a coenzyme for glutamate decarboxylase. So decrease in pyridoxal phosphate may also predispose to convulsions, especially in the neonates. So that's why pyridoxine supplementation is also one of the way to treat convulsions. Now, glutamate decarboxylase has also been secreted by beta cells of pancreas. The GABA that is made by glutamate decarboxylase is recycled by a process called GABA shunt, where a GABA undergoes catabolism to form succinate, and then that re-enters into TCA cycle. Like glutamate converted to GABA by decarboxylation, GABA removing amino group becoming succinate semialdehyde, and succinate semialdehyde becoming succinic acid or succinate, where NAD is reduced into NADHH plus, while succinate semialdehyde will be oxidized into succinic acid or succinate, and this succinate going into TCA cycle there. So that's how a GABA shunt will bring that. So succinate converted to fumarate, fumarate to malate, malate to oxaloacetate, oxalate combines with acetyl-CoA to make isocitrate into alpha-ketoglutarate forming glutamate, and glutamate will be converted back into GABA by glutamate decarboxylase, and that's how a GABA shunt goes. Now, enzyme glutamate decarboxylase is sensitive to drugs like hydrozides, and that's why hydrozides are used. Hydrolazine, example of hydrozides, hydrolazine. So it has got a vasodilatory. So these are the two special products that I have explained here in this lecture. Those are histamine and GABA. Thank you.